Assalamualaikum and a good day to our parents and friends. I am NURUL SHAHEIDA binti Abdul Aziz. Today, I present a review of Advanced in Antarctic Research for Anticancer Discovery, a comprehensive narrative review of marine animals from Antarctic environments as potential source of bioactive compounds against cancer cells. A big thanks to my supervisor, Dr. Sharifah Aminah Said Muhammad, who gave me a lot of guidance in completing my final year projects. First, let's begin with the introduction in Chapter 1, which consists of the background of the study. This Antarctic is the farthest south on the planet, and this continent is the coldest, driest, and windy, and more hostile. Microorganisms such as various marine animals are essential and must dominate most Antarctic ecosystem. Marine animals in the Antarctic have distinctive metabolic properties that can be adapted to the prospective use of biotechnology in various disciplines, particularly in the production of anti-cancer therapy. This study aims to review the potential anti-cancer cells from Antarctic marine animals and their mechanism of action interaction with cancer cells. The problem statement in this study shows that cancer therapy is a significant global health challenge with millions of new cases reported annually all over the world. Many Antarctic marine animal species remain poorly studied, making it challenging to identify and isolate bioactive compounds from this organism due to limited knowledge and research of their biology and physiology. Scientists find the study and gathering samples from this organism difficult because of the harsh and remote Antarctic environment. This review aims to identify the bioactive compounds produced by marine animals from the Antarctic for anti-cancer and harness the therapeutic potential as a cancer therapy. The objective of this study are to discuss the type of animals found in the Antarctic environment to produce anti-cancer. I was completing this objective in my FYP1. In my FYP2, I focus on two objectives only, which are to review the anti-cancer drug that has been discovered in Antarctic marine animals and to report on the anti-cancer mechanism of compounds discovered from Antarctic marine animals. The one objective covers one chapter of this article review. Next, I proceed to the literature review in Chapter 2 of this study. There are many species on this list of Antarctic animals in different habitats that can be found all over the Antarctic, such as the one-reefed albatross, Kenton penguin, southern elephant seal, Antarctic crane, scarabia seal, emperor penguin, blue whale, kitgum, leopard seal, marine invertebrate, and marine invertebrates. But in this literature review, I focus on animals that can produce anti-cancer, which are emerald rockcod fish, skunks, tunicates, spiroswan, nudibranch, and sea animals. The first animals that can produce anti-cancer are emerald rockcod tibernaci. This fish has developed specific protein adaptation in their bodies that keep their blood from freezing, which allows them to withstand the frigid water of the Antarctic. Next, the marine invertebrate that can produce anti-cancer is skunk K. wirelessa. These skunks produce antifreeze protein that stop ice crystal from forming inside their cell to survive in below freezing. Then the next skunk is L. apicalis. L. apicalis can survive in below freezing temperatures because they make antifreeze protein which stop ice crystal from forming inside their cell. For the tunicates, Epiphyclanicum is found in a variety of habitats, including deeper water and shallow coastal region. As a filter feeder, Epiphyclanicum had the ecosystem nutrient cycle by drawing in salt water and removing organic particles and microscopic organisms with their unique structure. Bryozoan, which are bibugula colonies, contribute to the overall biodiversity of the Antarctic marine ecosystem by providing other organisms with a substrate and a place to live. As a filter feeders, bibugula contributes to the cycling of nutrients by eating organic particles and plankton from the water column. Next, the marine invertebrates that can produce anti-cancer are nudibranch, Bihoxoni. These nudibranch have physiological adaptations that allow them to endure the bitterly cold marine environment of the Antarctic. Lastly, sea animals, Thermoctetis species. Thermoctetis species have specific compounds or proteins that help keep them from freezing or help them modify their metabolic process when the temperature drops. In Chapter 3, the diversity of marine life found in the world ocean present an almost unexplored opportunity for the discovery and creation of new drugs and therapies to fight cancer. Based on the article research conducted thus far, several anti-cancer compounds derived from marine life have been given to go ahead for clinical application, and a few more are presently undergoing clinical trial. The example of anti-cancer drug discovered in Antarctic marine animals are ankylansine in tibernaci, varolin B and entrin-methyl tetrahydrovarolin in K. wirelessa, discohabdin in L. apicalis, trabectedin in Epiphyclanicum, varistatin in B. bibugula, tisonconella actaloid in B. auxoni, and cytolytic peptides in Thermoctetis species. These anti-cancer therapies can treat various types of cancer such as leukemia, breast cancer, and lung cancer, also ovarian cancer. The anti-cancer compound discovered in Antarctic marine animals in this study have three main groups, which are peptides, alkaloids, and complex microcyclic lectin. The peptides group consists of ankylansine and cytolytic peptides. Next, the alkaloid group consists of varolin B and entrin-methyl tetrahydrovarolin, discohabdin, trabectedin, and isoquinoline alkaloids. Lastly, the complex microcyclic lectin consists of bryostatin. Research for new anti-cancer treatment according to different group benefit for further investigation into marine habitats as well as the characterization and screening of their inhabitants for bioactive compounds. Next, I will introduce the anti-cancer compound discovered in Antarctic marine animals. First, ankylansine is a peptide that modulates immune response, potentially enhancing the immune system ability to identify and eliminate cancer cells. Another anti-cancer agent is cytolytic peptide, which disrupts cellular membrane, specifically targeting and destroying cancer cells. These cohabdin are alkaloids that inhibit topoisomerase enzyme essential for DNA replication and cell division, leading to DNA damage and the death of cancer cells. Trabectedin contains tetrahydrofuran rings crucial for stabilizing the molecule, enhancing the DNA binding properties, thereby boosting its anti-cancer efficacy. Isoquinoline alkaloids, which are nitrogen-containing heterocyclic compounds, display various biological effects, including anticancer activities. Antimethyl tetrahydrovaline is a modified form of the valine B system, including additional saturation and a methyl group at a specific nitrogen atom in the ring structure. Both valine B and antimethyl tetrahydrovaline inhibit topoisomerase, a crucial enzyme for DNA replication and cancer cell cycle progressions. Lastly, biostatin targets proteokinase and enzyme cushion for quaternary cell growth, differentiation, and apoptosis, making it effective against specific type of leukemia, lymphoma, and solid tumors. The last objective in Chapter 4 is to explain the mechanism of anticancer compound from Antarctic marine animals, which focus on peptides, alkaloids, and complex microcyclic lectin. For the peptide, the first step is peptides target and bind to receptor or protein that are overexpressed on the surface of cancer cells. Next, positive charge interact with the negatively charged membrane of cancer cell, leading to membrane destabilizations. By blocking angiogenesis, peptides can starve the tumor of nutrient and oxygen and inhibit its growth. Immune system modulation of peptides can modulate the immune system to recognize and attack cancer cells. Lastly, peptides interfere with specific signaling pathway for cancer cell proliferation and survival. Next, the mechanism of alkaloids is the intercalation of alkaloids disrupt the DNA structure and function, inhibiting replication and transcription. Induction of apoptosis of alkaloids can induce apoptosis in cancer cell by activating intrinsic mitochondrial or intrinsic DUS receptor apoptotic pathway. Alkaloids inhibit the formation of new blood vessel and angiogenesis process that supply tumor with nutrients and oxygens. Antioxidative activity protects DNA from oxidative damage, will help to prevent mutation that can lead to cancer. The immunomodulatory roles of alkaloid enhance the activation and proliferation of cytotoxic T lymphocytes, which are critical for targeting and killing cancer cells. Lastly, I proceed to the mechanism of complex microcyclic lectins. The first is this anticancer compound interferes with cell cycle progressions, causing cancer cells to hide at specific checkpoints. Complex microcyclic lectins activate intrinsic apoptotic pathways by engaging the receptor. Example, trace on the cancer cell surface, leading to caspase activation and apoptosis. Inhibition of angiogenesis on complex microcyclic lectin inhibit the formation of new blood vessel angiogenesis process that tumor need for growth and metastasis. Complex microcyclic lectins inhibit key signaling pathway involved in cancer cell survival and proliferation and bind to tubulin, a process which the body block of microtubules and the stabilizing them. Modulation of immune response of complex microcyclic lectins can enhance the immune response against cancer cells by modulating the activity of immune cells such as T cells and macrophages. In conclusion, Antarctic marine animals represent a promising reservoir of bioactive compounds with potential application in cancer research. The comprehensive narrative review highlight the diversity of organisms inhabiting Antarctic environments and their unique adaptation, which can contribute to the production of bioactive molecules. These studies underscore the significant progress in identifying novel compounds from these organisms that exhibit anti-cancer properties through various mechanisms, including apoptosis induction, cell cycle arrest, and inhibition of metastasis. Developing environmentally friendly extraction techniques that minimize ecological impacts while maximizing compound yield is essential for the long-term viability of Antarctic bioreserve as source of anti-cancer agents. This article review has some of the recommendations. The first is, researchers must discuss the challenges faced in Antarctic research, such as extreme conditions, sample preservation, and ethical consideration to provide solutions and the best practice for conducting research in this unique environment. Researchers must focus on elucidating the molecular mechanism underlying the bioactivity of these compounds, optimizing extraction methods, and conducting preclinical and clinical trials to validate their efficacy and safety in cancer treatments. Lastly, more research is needed to elucidate the specific pathway involved in apoptosis inductions, cell cycle arrest, and inhibition of metastasis by these bioactive compounds, as safe as cancer therapy. That is all from me. Thank you.